Leonard to the table. Me and Martin actually studied together in UCD, so it's nice to be back.
Yes, indeed it is. Very nice to be back. Up until a couple of years ago, I worked for many years here in UCD and over at the Company Institute, so I have very fond memories of the place during that time and also during my degree. So today I'm going to talk to you about the Aryl Hydrocarbon Receptor and just give an introduction on how it's involved in many different processes, including the immune system. And then I'll just end the talk describing some of the work that we're carrying out at Public Health England, investigating hazard identification and characterization as part of a new strategy, a new research strategy within the Toxicology department. So the Aryl Hydrocarbon Receptor is a transcription factor and a member of this BHLH class family, and this family it's involved in development and environmental sensing. During the Metazoan evolutionary point, its function was mainly or predominantly in development. With the evolution of vertebrates, it adopted a ligand binding activity, and this is thought to be a control point for the regulation of detoxification enzymes such as CYP1A1. So, in terms of ligand binding, it has diverse groupings of chemicals which act as ligands, such as the halogenated aromatic hydrocarbons, and these include the dioxins, as you'll be very familiar with, but also the polycyclic aromatic hydrocarbons, and these include benzoylpyrene, for example. And then more recently, numerous endogenous ligands have been discovered for the Aryl hydrocarbon receptor. A lot of these have been identified as tryptophan metabolites. So not only can the Aryl hydrocarbon, like many other receptor transcription factors, it not only can bind agonists, but also cause different conformational changes within the protein, which in turn then cause different downstream effects on the genes that it regulates. So, following on from that point, then it is clear that there's very many ligands for the AHR, and there's also ligand-specific effects within the same system. If you apply different AHR ligands, you will get different signatures of AHR-dependent genes and also AHR-dependent responses. And this can be mediated through the interaction with different nuclear factors, the dimerization partners, and in turn then binding to potentially unconventional DREs or unique DNA sequences, or being controlled by being bound to different coactivators. So I'm sure you're all very aware of the signaling events which occur in the regulation of gene expression by the Aryl hydrocarbon receptor, but I'll just go through it briefly. So you have endogenous ligands which bind to the AHR complex, which then causes a translocation event, which then results in the loss of these dimerization partners and the binding of the AHR to ARNT, and this in turn then induces the transcription of CYP1A1, the prototypical enzyme or gene induced by the AHR. And there are also control points such as the AHR repressor protein. So when you get the induction of genes such as CYP1A1, these then act on the chemicals, the endogenous ligands, through hydroxylation events, and ultimately the aim would be to detoxify the chemicals. So there's one caveat or one of the caveats to that particular concept of detoxification. Again, I'm sure you're well aware of this, is the paradigm of the benzoylpyrene effect, where this process of inducing CYP1A1 by BAP, the enzyme itself actually turns the benzoylpyrene into a reactive metabolite, which then binds to DNA and is thought to be involved in mutagenesis. So that's probably one of the most studied toxicological mechanisms as a consequence of the AHR pathway. But there are a range of other functions of the AHR, including, as I mentioned, the xenobiotic response, but also it has effects on cell cycle and the estrogen receptor. It can have effects there, and also on the NRF2. And what I'll explain a little bit more is how it can also affect the immune system and both pro-inflammatory and anti-inflammatory effects. So one of the first studies looking at the effects of the Aryl hydrocarbon receptor was using a knockout mouse model, and the two main functions of, or the two main effects that they saw in those animals that survived was a decreased accumulation of lymphocytes in the spleen and lymph nodes, and also liver development. But also in terms of the toxicities induced by the AHR, a lot is known about this in human studies through exposures such as what has occurred with dioxin, for example, and included in that is immunosuppression. So just to delve down a little bit deeper in more recent studies looking at how the Aryl hydrocarbon receptor can specifically modulate the immune system and different inflammatory pathways. This recent paper in Nature investigated the differentiation of T cells and particular types of T cells as part of the adaptive immune response. And interestingly, they found in a model of murine encephalitis that depending on the ligand that was used, so an exogenous or dioxin exogenous ligand induced the differentiation of regulatory T cells, while the use of an endogenous ligand, these tryptophan metabolites, actually caused the differentiation of towards a Th17 profile, with a pro-inflammatory profile, as opposed to a more tolerance-based adaptive immune response. And also another study recently demonstrated the effect of the Aryl hydrocarbon receptor in the switching of T cell differentiation from one type to the other. So just another example in the skin, and an immune inflammatory path model in the skin, where they looked at Th2 dependent responses, and a treatment that is commonly used is coal tar, which contains AHR ligands. And they postulated this mechanism through which coal tar is having a protective effect. Firstly, through the regulation of proteins such as filaggrin, which is important for epidermal differentiation, but also through the regulation of an antioxidant response pathway, which was involved in the control of this pro-inflammatory transcription factor STAT6 through a phosphatase action. So there's just two or three examples there of how more in-depth knowledge about how the Aryl hydrocarbon receptor can actually affect the immune system. So this slide is just summarising some of these different effects and different pathways where the Aryl hydrocarbon receptor can have its effects. So in terms of the activities that we've started looking at Public Health England, and the interest within the toxicology department in one particular program is to investigate hazards within respiratory pollutants, such as diesel exhaust material and indoor space heating, and a lot of those active substances that could be considered as detrimental to human health are AHR ligands. So we wanted to focus on that in some way. So the way we began this was to take an in vitro model, and I must say that this is part of this piece of work I'm going to show is part of a larger strategy to include in vivo studies and also epidemiological investigations into hazards of pollutants and Aryl Hydrocarbon Receptor ligands. So we started off looking at primary bronchial epithelium from different donors and exposing them to the prototypical AHR ligands, pollutants, benzoate-pyrene, and then we carried out some RNA-seq analysis, and this lower heat map just summarizes the gene changes that we observed across a dose response. So when we used ingenuity pathway analysis then to just summarize what kind of responses we were getting, and within the top 10 regulated pathways we identified those involved in the adaptive immune response. So if we looked at a different aspect of IP analysis of the same data, where you can have significance, where we're looking at regulators of the gene expression, predicted regulators or associated regulators of the gene expression. So you have significance on this axis, and then direction of change or predicted direction of change. And unsurprisingly, you see benzoate-pyrene and cigarette smoke and also TCD as positive regulators of this predicted positive regulators of just gene expression. But you also see estradiol. Again, that may not be that surprising because of its broad effect on different pathways. And also you see an effect here on HIF1 alpha, and again this could possibly be explained as a negative regulator, or it being associated as being negatively regulated because the AHR uses the same binding partner, so it could be a cool effect. So in terms of just characterizing this a little further, we took some of the genes that we identified from the RNA-seq analysis and looked for the effect or the effect of other polycyclic aromatic hydrocarbons commonly found in pollutant material with different degrees of prototypic CYP1 activity. And the long and short of this is that you do see a parallel effect of immune response related genes being induced at the same level, at same representative level as benzoate-pyrene, or relative level as benzoate-pyrene, and you also see an oxidative stress response. So we've brought this a little bit forward by looking at some of these immune responses in diesel exhaust particulate material, and again we see similar effects of pro-inflammatory cytokine gene expression. So just to summarize then this data, I think the message I would like to get across is that the Aryl hydrocarbon receptor has diverse functions, and it also has a broad array of ligands and ligand-specific effects. And in the context of, for example, what Alice showed earlier on, trying to tease out individual responses from chemicals, I think we have to consider that the activation of one pathway can also affect many, many other pathways, and teasing out that may not be a very straightforward thing to attempt easily. So also the effect of the Aryl Hydrocarbon Receptor on the immune system is wide-ranging, and consideration, this is an obvious statement, but consideration of the AHR as part of an overall approach to evaluate chemicals and also particles and safety testing strategies is important to build weight of evidence for mode of action and or AOP approaches. And also, in terms of moving forward with the work that we're carrying out in PHE, we also consider that analysing these toxic effects in disease-modifying models is also important. And in terms of the ones that we would proceed with, these would be allergy and asthma-based studies. So that's how we see. And the reason partially for that, because a lot is known about the effect of air pollution, for example, on the exacerbation events of asthma. But there is there's quite a bit of controversy about whether air pollution and polycyclic aromatic hydrocarbons, whether they're involved in actual development of the disease. So that's something that we would like to pursue in the future, using these and other methods. So thank you.